Good afternoon. Welcome to the first day of the immune system unit. Seemed only appropriate to have the immune system be the first body system we cover. And today what we'll be looking at is the general structure of viruses and then some of the specifics of how it is that SARS CoV-2, here's that common World Health Organization picture that you see on the news a lot of SARS CoV-2, the cause of COVID-19. We'll look at how exactly that invades our cells. We'll also kind of look a little bit at flu and HIV as well, just so we can kind of see what some viruses have in common. We will then also look, and this is kind of one of the most interesting parts to me, is how I'm able to get over it. So let's say that my cells, we have the cells that get infected by SARS CoV-2 are a type of lung cell. You don't have to know the name of it for my PreAP students, but it's called a type 2 pneumocyte. Pneumo means lung, like pneumonia. So it turns out that SARS CoV-2 infects type 2 pneumocytes, lung cells. And we're going to look at what happens whenever I get an infection, and then what does my body do in retaliation so that I eventually get better, hopefully, if I happen to get sick. So we'll start with the basic structure of a virus. Viruses have two things in common across the board. They have to have genetic material, DNA or RNA, and they have to have a protein coat. That protein coat might sometimes be called a capsid. So over here, I'm always drawing my RNA as single-stranded. It's actually possible to have double-stranded RNA. But flu has a piece of RNA, and then it's got a green protein coat or capsid around it. Same thing with HIV. It's got some RNA and then a protein capsid around it. Over here, SARS CoV-2, it sort of has like chunks of proteins. It functions as one protein, but it's actually like pieces of a protein, almost like I have a whole bunch of bricks, but they still function as one single wall. And then I have my RNA. I don't know how well you can see it, but I have some brown RNA in there too, just like I have RNA over here in these. So let's look at HIV really quickly. So we'll have some sort of protein sticking off on the outside of a virus. In HIV, it's this glycoprotein structure right here. I'll zoom in a bit. And those glycoproteins are there to help the virus actually get inside of a cell. So you'll notice these glycoproteins, they just have a specific shape. I'm not saying they look exactly like this, but they have a shape. Same thing over here with flu. These flu viruses have a couple of proteins on them. They have a hemagglutinin and a neuraminidase. You don't have to know the names. But what's significant is if I look at flu, let me pull up my flu diagram here. If I look at flu, hemagglutinin starts with an H, neuraminidase starts with an N. You might have heard of like H1N1 flu or H5N1 flu is H1N1 was the one that caused like the 1918 Spanish flu epidemic and pandemic, excuse me. An epidemic is when it's somewhat local, like if there was an infection and it was widespread throughout Texas, but once it starts to be more and more global, we call it a pandemic. So COVID-19 is a pandemic because it's found in however many countries it's in now. So anyway, back to flu. There are different versions of hemagglutinin and different versions of neuraminidase, and we just number them. And so if you have H1N1 flu or H5N1 flu, hopefully you don't have H5N1, that one's really bad. But that's basically naming it based off of the proteins that are on the outside. Okay, so now let's back up, zoom back out. Sorry, this is just like my phone and a room in my house, so it's not 100% professional. Okay, so now what we want to do is we want to look at SARS-CoV-2 versus like HIV. So back over here with HIV, you'll notice this particular cell. You don't have to know the name of it, but it's a helper T cell. And this helper T cell has proteins on it with a certain shape. HIV has these glycoproteins with a certain shape. What you'll notice is that shape fits that protein. You don't have to know the name of it, but that particular protein is called CD4. So it turns out cells, like helper T cells aren't the only cells that have CD4 proteins on them, but cells with CD4 on them are able to get infected by HIV because that fits that sort of like a lock and a key. There's another protein, I'm just doing this to be complete. You don't have to sweat it, but there's another protein called CCR5 that's also needed. So the way that HIV actually infects a cell is it grabs this first protein, and then that first protein kind of hands it off to the second protein, and then HIV pulls itself inside. So these guys act as like grabbers and then eventually pullers to pull themselves inside of a cell. Well, now let's look at SARS CoV-2. There was a SARS around 2002, throughout the 2002-2003 time period that was very deadly. That was SARS CoV-1, and this is SARS CoV-2. Well, you see on the news these spike proteins that they talk about a lot. On the WHO picture, they're red triangles, so I made mine red triangles also. That spike protein is what grabs onto a protein on this cell. So this cell we're just going to call a lung cell. But again, it's actually what's called a type 2 pneumocyte. But those are found in your lungs, so I'm just going to call it a lung cell. And these lung cells have this particular protein on them called ACE2. Don't worry about it, it's a protein on there. Okay, so that and that have a specific shape that's complementary. So when SARS-CoV-2 grabs onto this, that's the beginning of it being able to pull itself inside of a cell. So it turns out there's another protein inside that the cell has that also helps for it to eject its genetic material inside. And that's actually where we think we might have a drug that might be a treatment for SARS-CoV-2. They've actually even got a journal article about it. We'll get to it eventually. But the journal article hasn't even come out yet. It's coming out in the journal Cell on April 16th. But it's basically talking about this other protein that's needed for SARS-CoV-2 to be able to enter into a cell. And if we can block that protein, we already have the ability to do that. We might be able to kind of treat SARS-CoV-2. So anyway, in the meantime, back over here, these little red triangles grab onto ACE2, and then the virus is able to pull itself inside. Well, that sucks, because once that happens, now that piece of RNA will be red. So who is it that reads RNA in our cells and converts it into protein? Ribosomes. So it hijacks my cells, and if there's a piece of RNA there, ribosomes will read it. So ribosomes start reading that, and they build the proteins that it says to build. So it starts building the orange, the blue, the red, because that's what it's told to do. So it's building all these various proteins that you see in that picture. Now you can probably only see the red ones, but there's some orange and yellows in there also. So I don't have a yellow, so I'm using orange and blue. And then what will happen is it tells the cell, my lung cell, hey, ship this stuff up to the surface of the cell. And your cell's like, all right. And so now we start shipping all these proteins to the surface of the cell, like this. And now it will then tell the cell like, hey, RNA, start making copies of me. It has this RNA-dependent RNA polymerase, so it can basically read RNA and convert it into RNA. You might remember when we were talking about the cell cycle in the S phase, we had DNA polymerase that read DNA and made copies of it into more DNA. So now we also have the RNA being copied, like this. Well, we'll also build the proteins, just like this. So what you'll see is we're now building all the pieces to making the virus. To make this virus, we need orange, blue, red. We've got those. We need the RNA and we need the green proteins. So now all we do is we ship it out. So it's just like exocytosis from the second or third unit of study that we had. Now my cell looks like this. It just starts mass producing SARS-CoV-2 viruses. So they just bud off just like this. See, you got red. And so now I'm basically just turning this lung cell, this pneumocyte, into a virus factory. Like this. And that'll bud off, and eventually once it pinches off, then it's a new virus, not a new cell. Viruses are considered non-living, so I shouldn't call them cells. And then this virus will just go and infect the next cell that has an ACE2 protein on the outside coat. So my cell just became a virus factory. Okay, well, what's a cell to do? I have to somehow, if I'm a cell and I'm invaded, I have to somehow warn everybody around me that I'm invaded. So the way that my immune system generally works, and in the AP class we kind of go into more detail. I might shoot some immune system videos for the AP level 2 where we kind of fill in a few of these blanks. But the basic way that the immune system works is, let's say that we're Timber Creek, purple, purple. So let's say that purple cells are Mr. Cotton cells. So this is a Mr. Cotton lung cell, and this is a Mr. Cotton kidney cell. Well, here's a white blood cell. A white blood cell is from my immune system, and apparently it does this. And what it'll do is it'll go along and it'll check surface proteins. It'll check glycoproteins and glycolipids on my cells and be like, everything's cool. Everything's cool. If it bumps into a bacterium, it's like, not cool. Then it'll do phagocytosis and gobble up that bacteria, rip it apart, and spit out the bits. Okay, great. However, what if I've got a Mr. Cotton lung cell and I've got another Mr. Cotton lung cell? Okay, everything's cool, everything's cool. But what if it's not cool? What if inside of here, viruses got inside and they're now infecting me, and this is now becoming a virus factory? How could you tell? Picture it like this. Here's me. And then here is me infected by a virus. From the outside, you can't tell the difference hardly. So how are we going to be able to tell? Well, it turns out we have these cells, and you don't have to worry about the name, but they're a type of immune system cell called cytotoxic T cells. We have several different types. We'll probably mention helper T cells and cytotoxic T cells. I don't know that we'll mention any other types of T cells like Tregs and stuff like that. So a cytotoxic T cell has a receptor, and it's also got another protein. And what it'll do is it's checking this protein on my own cells called MHC. Now, I know that we're throwing out a whole lot of terms, but here's an MHC, and just for accuracy, it's called MHC class 1, so MHC1. And MHC1 proteins, their job is they're constantly going to the middle of the cell, grabbing whatever's floating around in the cytoplasm, whatever proteins are floating around, and just holding them for the outside world to see. Again, our problem is this. If I am a Mr. Cotton lung cell, and I'm infected with a virus, viruses are replicating inside of me. When the white blood cell comes and checks me from the outside, I look okay. So how can I tell him, hey, I'm not okay? I do that with MHC1. So normally what will happen is MHC1 will go and grab Mr. Cotton proteins that are floating around in there. They're purple because, again, Mr. Cotton stuff is purple, we'll say. So my MHC1 will be holding out a piece of Mr. Cotton protein. My cytotoxic T cell will be checking, and it'll be like, all right, everything checks out. But if I have viruses growing inside of me, replicating inside of me, then I might have one of these spike proteins. So MHC1 just goes to the middle of the cell, just grabs something, goes to the edge of the cell and holds it out there. Nothing? All right, great. Grab something, holds it out there. Nothing? Okay, great. But eventually it's going to grab something that's not okay. It's going to grab a viral protein and hold it out there. A cytotoxic T cell that happens to have a receptor that fits that. Once this cytotoxic T cell bumps into that, this cytotoxic T cell now knows that I'm sick. It's a little bit like this. If I'm invaded, back to being invaded by this, if I could like get a piece of it and hold it on the outside of me, it's like, yeah, I'm wearing Timber Creek purple, but look at this. This is what's inside of me, everybody, and it's not purple. If it was purple, you'd leave me alone. So on my MHCs, I show you this and you're like, cool, you're supposed to have purple. But if instead I hold out this, you're like, that's not cool. And so now you have to die. So what cytotoxic T cells will do is we're going to focus on two ways that they can kill this cell. One is called Perforin. Enzymes end in ACE, they're proteins that speed up chemical reactions. If something ends in IN or EN, it's probably a protein that's not an enzyme. So this is a protein that does perforations, pokes holes. You might think of like perforated paper. Like I can tear across the top of this. They're little bitty holes, perforations, so that it tears easily. So they can shoot perforin at my Mr. Cotton cell. Now, this is not a healthy one, so we're trying to kill the cell. All right, so this cytotoxic T cell shoots perforin, and that literally pokes holes in the surface of the cell. The pictures I'm going to show you are actually from a different system that pokes holes called complement, but it's the same idea. That's the surface of a cell with a bunch of holes poked in it. And so eventually what will happen is diffusion. Things move in, things rush out, osmosis, water rushes in or out, and the cell explodes. So to summarize, the cytotoxic T cell will shoot the unhealthy cell, and the unhealthy cell explodes. Now, the second thing that it can do is it can turn on, that just says on if you can't see it very well on your screen, it turns on the genes for apoptosis. Apoptosis. Apoptosis is a sequence of genes in all nucleated cells, all cells with a nucleus, have a gene sequence for apoptosis. And apoptosis is programmed cell death. So it's basically the self-destruct button. So right now, my kidney cells have the genetic code for apoptosis, for destroy yourself. But that code is currently off, right? Just like a nuclear launch code or whatever, like, oh, we could launch a missile, but I haven't pushed the button yet. So what can happen is this cytotoxic T cell can send proteins in and turn those genes on, and now the cell will kill itself. So this is the equivalent of a cytotoxic T cell going up to this lung cell and saying, hey, kill yourself. And the lung cell's like, all right. Now, at first that seems terrible. It's like, well, my cells are dying, but I just shut down a virus factory. So if I can get ahead of this and shut down all the virus factories, maybe I can grow some new type 2 lung cells. Or if they were kidney cells, maybe I can grow some new kidney cells, right? If I get a cut on my arm, I can just clear out the deadies and then put in some new ones, and now I'll be as good as new in however many days. So that's what we're hoping for, is that when cells get sick, they can tell on themselves and say, hey, you ought to kill me, and then we either shoot it full of perforin or we turn on the cell suicide sequence. Again, that's just the cell killing itself, not the organism. So the cliffhanger that will be addressed in the next video is, how did this cytotoxic T cell have a receptor that fit that spike protein? Right? My body has never seen SARS CoV-2. In fact, you've heard that SARS CoV-2 is a novel virus. That means new. So humans, until recently, haven't seen it. So how do we already have receptors for it? For example, the flu of 2030 that will have different proteins on the outside, I actually already have some receptors for it, even though that version of the flu doesn't exist. So how do I do that? The last analogy I'll do with that is, I might have even used this analogy in class, is pretend that somebody's going to ship me a key. They haven't even made the key yet. They're going to ship me the key in a year, and I have to have a lock that fits it. The second that key arrives, I have to have a lock that fits that key. So how do I do that? How do I have a receptor, a lock for a protein, a key, if that's brand new and I've never seen that before? So that's what we'll talk about in the next video. We'll also talk about vaccines in the next video. Thanks for coming out.